there are another 2 videos soon to follow. So these videos are about the nervous system, which is one of the most important systems in the body because it regulates sensory input, sensory integration, and motor output. Pay attention to the integration aspects of the nervous system. In other words, its programming, and also how it uses electrical impulses to carry and communicate information to the brain. This is a crash course in knowing your body, its anatomy and physiology. Enjoy the following video from Crash Course. There are 2 videos in this topic. Thank you for watching, and be sure to watch the next part 1K.
This morning was a typical morning for me. I woke up thinking about that dream that I keep having about the guy in the sloth suit, and then I got dressed because I was cold, and then I made some toast with butter because I was hungry, and then I let the dog out because she was whining and staring at me, and then I made some tea, but I let it cool off before I drank it because it burned my mouth yesterday. In addition to being just part of my morning ritual, all of these actions are examples of what my nervous system does for me. The weirdo dream, the sensation of cold air and hot tea, deciding what to put on the toast, going to the door, the sound of the dog, all of that was processed and executed by electrical and chemical signals to and from nerve cells. You can't oversell the importance of the nervous system. It controls all the things. All your organs, all your physiological and psychological reactions, even your body's other major controlling force, the endocrine system, bows down before the nervous system. There is no you without it. There's no me without it. There's no dogs without it. There's no animals. There's no things, there's things. It's important. That's why we're dedicating the next several episodes to the fundamentals of the nervous system, its anatomy and organization, how it communicates, and what happens when it gets damaged. This is Mission Control, people. Even though pretty much all animals, except super simple ones like sponges, have a nervous system, ours is probably the most distinctive feature of our species. From writing novels to debating time travel to juggling knives, all of your thoughts and actions and emotions can be boiled down into 3 principal functions: sensory input, integration, and motor output. Imagine a spider walking onto your bare knee. The sensory receptors on your skin detect those 8 little legs. That information is your sensory input. From there, your nervous system processes that input and decides what should be done about it. That's called integration. Like, should I be all zen about it and just let it walk over me? Or should I not be zen and freak out and run around screaming, "Spider!" Your hand lashing out to remove the spider and maybe your accompanying banshee scream is the motor output, the response that occurs when your nervous system activates certain parts of your body. As you can imagine, it takes a highly integrated system to detect, process, and act on data like this all the time. And when we talk about the nervous system, we're really talking about several levels of organization, starting with 2 main parts, the central and peripheral nervous systems. The central nervous system is your brain and spinal cord, the main control center. It's what decided to remove the spider and gave the order to your hand. Your peripheral nervous system is composed of all the nerves that branch off from the brain and spine that allow your central nervous system to communicate with the rest of your body. And since its job is communication, your peripheral system is set up to work in both directions. The sensory, or afferent division, is what picks up sensory stimuli, like, hey, there's an arachnid on you, and slings that information to the brain. Your motor, or efferent division, is the part that sends directions from your brain to the muscles and glands, like, hey, hand part, how about you do something about that spider? The motor division also includes the somatic, or voluntary nervous system that rules your skeletal muscle movement, and the autonomic, or involuntary nervous system that keeps your heart beating and your lungs breathing and your stomach churning. And finally, that autonomic system, too, has its own complementary forces. Its sympathetic division mobilizes the body into action and gets it all fired up like, spider! While the parasympathetic division relaxes the body and talks it down, like, it wasn't a black widow or anything, you're fine, breathe. So that's the organization of your nervous system in a nutshell. But no matter what part you're talking about, they're all made up of mainly nervous tissue, which you'll remember is densely packed with cells. Maybe less than 20% of that tissue consists of extracellular space. Everything else cells. The type of cells you most likely heard of are the neurons, or nerve cells, which respond to stimuli and transmit signals. These cells get all the publicity. They're the ones we're always thanking every time we ace an exam or think up a snappy comeback to an argument. But these wise guys really account for just a small part of your nervous tissue because they are surrounded and protected by gaggles of neuroglia, or glial cells. Once considered just the scaffolding or glue that held neurons together, we now know that our different glial cell types serve many other important functions, and they make up about half of the mass of your brain, outnumbering their neuron colleagues by about 10 to 1. Star-shaped astrocytes are found in your central nervous system and are your most abundant and versatile glial cells. They anchor neurons to their blood supply and govern the exchange of materials between neurons and capillaries. Also in your central nervous system are your protective microglial cells. They're smaller and kind of thorny looking and act as the main source of immune defense against invading microorganisms in the brain and spinal cord. Your ependymal cells line cavities in your brain and spinal cord and create, secrete, and circulate cerebrospinal fluid that fills those cavities and cushions those organs. And finally, your central nervous system's oligodendrocytes wrap around neurons, producing an insulating barrier called the myelin sheath. Now, over in your peripheral nervous system, there are just two kinds of glial cells. Satellite cells do mainly in the peripheral system what astrocyte cells do in the central system. They surround and support neuron cell bodies, while Schwann cells are similar to your oligodendrocytes in that they wrap around axons and make that insulating myelin sheath. So don't sell your glial cells short. They're in the majority cell-wise, but of course when it comes to passing tests and winning arguments, most of the heavy lifting is done by the neurons. And they're not all the same. They're actually highly specialized, coming in all shapes and sizes, from tiny ones in your brain to the ones that run the entire length of your leg. But they do all share three super cool things in common. Number one, they're some of the longest lived cells in your body. There's a lot of debate right now about whether you're actually born with all the neurons you'll ever have, but some research suggests that at least in your brain's cerebral cortex, your neurons will live as long as you do. Cool fact number two, they are irreplaceable. It's a good thing that they have such longevity because your neurons aren't like your constantly renewing skin cells. Most neurons are amitotic, so once they take on their given roles in the nervous system, they lose their ability to divide. So take care of them. And number three, they have huge appetites. Like a soccer-playing teenager, neurons have a crazy high metabolic rate. They need a steady and abundant supply of glucose and oxygen, and about 25% of the calories that you take in every day are consumed by your brain's activity. Along with all these wonderful qualities, your neurons also share the same basic structure. The soma, or cell body, is the neuron's life support. It's got all the normal cell goodies like a nucleus, DNA, mitochondria, ribosomes, cytoplasm. The bushy branch-like things projecting out from the soma are dendrites. They're the listeners. They pick up messages, news, gossip from other cells and convey that information to the cell body. The neuron's axon, meanwhile, is like the talker. This long extension or fiber can be super short or run a full meter from your spine down to your ankle. We've got a few different axon layouts in our body, but in the most abundant type of neuron, the axons transmit electrical impulses away from the cell body to other cells. For us students of biology, it's a good thing that nerve cells aren't all identical because their differences in structure are one of the ways that we tell them apart and classify them. The main feature we look at is how many processes extend out from the cell body. A process, in this case, being a projecting part of an organic structure. 99% of all your neurons are multipolar neurons with 3 or more processes sticking out from the soma, including one axon and a bunch of dendrites. Bipolar neurons have 2 processes, an axon and a single dendrite, extending from opposite sides of the cell body. They're pretty rare, found only in a few special sensory places like the retina of your eye. Unipolar neurons, on the other hand, have just one process and are found mostly in your sensory receptors. So if you ever find yourself probing around someone's nervous tissue, remember these 3 terms to help you figure out what you're looking at. But because we're talking about physiology here as well as anatomy, we have to classify these cells in terms of their function, and that basically comes down to which way an impulse travels through a neuron in relation to the brain and spine. Our sensory, or afferent neurons, pick up messages and transmit impulses from sensory receptors in, say, the skin or internal organs, and send them toward the central nervous system. Most sensory neurons are unipolar. Motor, or efferent neurons, do the opposite. They're mostly multipolar and transmit impulses away from the central nervous system and out to your body's muscles and glands. And then there are interneurons, or association neurons, which live in the central nervous system and transmit impulses between those sensory and motor neurons. Interneurons are the most abundant of your body's neurons and are mostly multipolar. Okay, it's applied knowledge time. Let's review everything we've learned so far in terms of that spider on your knee. Those 8 creeping legs first activate your unipolar sensory neurons in the skin on your knee when they sense something crawling on you. The signal travels up an axon wrapped in Schwann cells and into your spinal cord, where it gets passed on to several multipolar interneurons. Now, some of those interneurons might send a signal straight down a bunch of multipolar neurons to your quadriceps muscle on your thigh, triggering you to kick your leg out before you even know what's going on. Other interneurons will pass that signal to neurons that carry it up your spinal cord to your brain. That's where your body first recognizes that thing as a spider, and the connections between neurons interpret and split the signal so that you can either scream and start swinging your arms wildly about, or remain calm and with dignity remove the spider from your person. It's all based on the connections between neurons, which brings me to a whole new question. How? How in the name of Jean-Martin Charcot do nerve cells use chemistry and electricity to communicate with each other? It's one of the most stupefyingly awesome and complicated aspects of your nervous system and basically of all life, and it is what we will cover in our next lesson. Today, you learned how sensory input, integration, and motor output of your nervous system basically rules your world. We talked about how the central and peripheral systems are organized and what they do, and looked at the role of different glial cells in nervous tissue function. We also looked at the role, anatomy, and function of neuron types in the body, both structurally and functionally, and how everything plays out when you find a spider crawling on your skin. Thank you for watching, especially to all of our Subbable subscribers who make Crash Course possible for themselves and for the whole rest of the world. To find out how you can become a supporter, just go to Subbable.com. This episode was written by Kathleen Yale, the script was edited by Blake DiPestino, and our consultant is Dr. Brandon Jackson. It was directed by Nicholas Jenkins and Michael Aranda, and our graphics team is Thought Cafe. What if everything you did, and thought, and felt could be communicated by pushing a button? It'd be like using the world's simplest app, one that just sends out a little ping, always at the same volume and length, to communicate everything from "it sure is cold in here" to "I love churros" to "boy, I sure would like to breathe sometime soon." Well, that is actually exactly how your neurons send all the impulses responsible for every one of your actions, thoughts, and emotions. When a neuron is stimulated enough, it fires an electrical impulse that zips down its axon to its neighboring neurons. But they've only got one signal that they can send, and it only transmits at one uniform strength and speed. What they can vary is the frequency or number of pulses, like this is distinct from this. And your brain can translate these signals, reading them like binary code, organizing them by location, sensation, magnitude, and importance, so that you know the difference between "turn up the thermostat" and "oh my gosh, I'm on fire." That buzz, that nerve impulse, is called the action potential. It's one of the most fundamental aspects of anatomy and physiology, and really life in general. It's happening inside of you right now, and we want to make sure that you understand what all that buzz is about. Before we delve into how neurons communicate, we gotta first understand a little bit of our old friend electricity. Basically, think of your body as a sack of batteries. No, I mean, you don't look like a sack of batteries. I'm just saying that your body as a whole is electrically neutral with equal amounts of positive and negative charges floating around, but certain areas are more positively or negatively charged than others. And because opposite charges attract, we need barriers or membranes to keep positive and negative charges separate until we're ready to use the energy that their attraction creates. In other words, we keep them separated to build potential. A battery, just sitting on its own, has both a positive and a negative end, and the potential to release energy, but it doesn't do anything until it's hooked up to a flashlight or a phone or a kid's toy that lets those charges move toward each other, on the way converting electricity into light or sound or children's laughter. In much the same way, each neuron in your body is like its own little battery with its own separated charges. It just needs an event to trigger the action that brings those charges together. If you're thinking that this sounds more like engineering than anatomy, that might not be a bad thing. It might even help to think of your neurons in the same terms an electrician might use. Voltage, for example, is the measure of potential energy generated by separated charges. It's measured in volts, but in the case of your body, we use millivolts because it's a pretty small amount. In a cell, we refer to this difference in charge as the membrane potential. The bigger the difference between the positive and negative areas, the higher the voltage and the larger the potential. And just like there's voltage in your body, there's also current, the flow of electricity from one point to another. The amount of charge in a current is related both to its voltage and its resistance. Resistance is just whatever's getting in the way of the current. Something with a high resistance is an insulator, like plastic, and something with a low resistance is a conductor, like metal. Now, when we talk about these concepts in terms of U, we're typically talking about how currents indicate the flow of positively or negatively charged ions across the resistance of your cell's membranes. And again, these membranes separate the charges, so they're what provide the potential to convert the electricity into something useful. Okay, now that we got electricity 101 down, let's see how it works inside your nervous system. A resting neuron is like a battery just sitting in that sack that is you. When it's just sitting there, it's more negative on the inside of the cell relative to the extracellular space around it. The difference is known as the neuron's resting membrane potential, and it sits at around negative 70 millivolts. Where do those charges come from? Outside of a resting neuron, there's a bunch of positive sodium ions floating around, just lingering outside the membrane. Inside, the neuron holds potassium ions that are positive as well, but they're mingled with bigger negatively charged proteins. And since there are more sodium ions outside than there are potassium ions inside, the cell's interior has an overall negative charge. When a neuron has a negative membrane potential like this, it is said to be polarized. Now, these ions didn't just show up in this arrangement on their own. This is all orchestrated by one of the most important bits of machinery in your nervous system, the sodium-potassium pump. This little protein straddles the membrane of the neuron, and there are tons of them all along the axon. For every 2 potassium ions it pumps into the cell, it pumps out 3 sodium ions. This creates a difference in the concentration of sodium and potassium, and a difference in charges, making it more positive outside the neuron. This difference is an electrochemical gradient, and you probably know enough about biology by now to know that nature hates gradients. It wants to even out all of those inequalities in concentration and in charge to restore balance. But the only way to even out that gradient is for the ions to pass across the membrane. Thankfully, the sodium-potassium pump isn't the only way in or out of the cell. The membrane is also riddled with ion channels, large proteins that can provide safe passage across the membrane when their respective gates are open. And these gates open and close for different reasons depending on their structure and purpose. Most are voltage-gated channels, which open at certain membrane potentials and close at others. For example, sodium channels in your neurons like to open around negative 55 millivolts. But some others are ligand-gated channels. They only open up when a specific neurotransmitter like serotonin or a hormone latches onto it. Then we also have mechanically gated channels, which open in response to physically stretching the membrane. In any case, when the gates do open, ions quickly diffuse across that membrane down their electrochemical gradient, evening out the concentrations and running away from other positively charged ions. This movement of ions is the key to all electrical events in neurons, and thus is the force behind every single thing we think, do, and feel. Of course, not all of your body's electrical responses are the same, and neither are the flows of ions going in and out of your neurons. If only a few channels open and only a bit of sodium enters the cell, that causes just a little change in the membrane potential in a localized part of the cell. This is called a graded potential. But in order to send long-distance signals all the way along an axon, you need a bigger change, one big enough to trigger those voltage-gated channels. That is an action potential. And your best bet for making that happen is to depolarize that resting neuron. I mean, cause a big enough change in its membrane potential that it'll trigger the voltage-gated channels to open. It all starts with your neuron sitting there at resting state. All of the ion channels are closed and the inner voltage is resting at negative 70 millivolts. And then something happens. Some environmental stimulus occurs, say like a spider brushes up against a tiny hair on your knee, triggering those sodium channels to open, increasing the charge inside the membrane. Now, the stimulus and the resulting change have to be strong enough to cross a threshold for the true action potential to kick in, and that threshold is about negative 55 millivolts. Remember that number, because this is an all-or-nothing phenomenon. If the stimulus is too weak and the change doesn't hit that level, it's like a false alarm. The neuron just returns to its resting state. But kind of like Doc Brown hitting 1.21 gigawatts in the DeLorean, once it hits that threshold, you're not going to travel in time, but you are going to see some serious action potential. At that threshold, the voltage-gated sodium channels open, and there are tons of these, so all of the positive sodium ions rush in, making the cell massively depolarized. So much so that it actually goes positive, up to about positive 40 millivolts. This is action potential in action. It's really just a temporary reversal of a membrane potential, a brief depolarization caused by changes in currents. And unlike graded potentials, which are small and localized, an action action potential kicks off a biological chain reaction which sends that electrical signal down the axon. Because each of your neurons has lots of voltage-gated sodium channels, so when a few in one area open, that local current is strong enough to change the voltage around them, and that triggers their neighbors, which triggers the voltage around them, and so on down the line. As soon as all that's underway, the process of repolarization kicks in. This time the voltage-gated potassium ion channels open up, letting those potassium ions flow out in an attempt to rebalance the charges. If anything, it goes too far at first, and the membrane briefly goes through hyperpolarization. Its voltage drops to -75 or so millivolts before all of the gates close and the sodium-potassium pumps take over and bring things back to their resting level. Now, when part of an axon is in the middle of all this and its ion channels are open, it can't respond to any other stimulus, no matter how strong. This is called the refractory period, and it's there to help prevent signals from traveling in both directions down the axon at once. So that is the surprisingly simple app that your nervous system uses to let you experience the world. And because the voltages in this process are always pretty much the same, the initial threshold around -55 millivolts and the peak depolarization at 40 millivolts, your neurons only communicate in a single monotone buzz. It doesn't matter if it's a spider on your knee or an elephant, a paper cut or a stab wound, the strength of that action potential is always the same. What does change is the frequency of the buzz. A weak stimulus tends to trigger less frequent action potentials, and that includes if the stimulus is coming from you, like your brain telling your muscles to perform some task. If I need to do something delicate, like pick up an egg, the signal is low frequency. But a more intense signal, like trying to crush a can, increases the frequency of those action potentials to tell your muscles to contract harder, and the message turns into something that you can't ignore. Action potentials also vary by speed or conduction velocity. They're fastest in pathways that govern things like reflexes, for example, but they're slower in places like your glands, guts, and blood vessels. And the factor that affects a neuron's transmission speed the most is whether there's a myelin sheath on its axon. Axons coated in insulating myelin conduct impulses faster than non-myelinated ones, partly because instead of just triggering one channel at a time in a chain reaction, a current can effectively leap from one gap in the myelin to the next. These little gaps are the delightfully named nodes of Ranvier, and this kind of propagation is known as saltatory conduction, from the Latin word for leaping. But what happens when an action potential hits the end of its axon and is ready to do more than leap and jump all the way to another neuron? That you will find out next time. Today, you learned how your body is kind of like a big bag of batteries, and how ion channels in your neurons regulate this electrochemistry to create an action potential from resting state to depolarization to repolarization and a brief bout of hyperpolarization. Thanks for watching, especially to all of our Subbable subscribers who make Crash Course possible for themselves and for everyone else. To find out how you can become a supporter, just go to Subbable.com. This episode was written by Kathleen Yale, the script was edited by Blake T. and our consultant is Dr. Brandon Jackson. It was directed by Nicholas Jenkins and Michael Aranda, and our graphics team is Thought Cafe. One more thing before you leave. We like Crash Course a lot, and we hope that you like Crash Course a lot, but I kind of feel like Crash Course is only useful for a certain segment of the population. Like, once you get to a certain age, then it's good, and then forever, it can be helpful to people. But younger people, not so much. And so, we are creating Crash Course Kids, hosted by Sabrina Cruz from Nerdy and Quirky. Crash Course Kids will start out focusing on 5th grade science, but we'll keep expanding to other topics as the channel grows. Sabrina will be talking about food chains and gravity and how the sun works and how plants eat and why flamingos are pink and many other topics. Oh, and another note, teachers, you can rest assured that we've got you covered. There will be info about the standards we've used to make sure that we're doing our very best to help you out. So if you are a teacher, or you know a teacher, or you know a child, or you know someone who has a child, or you've ever seen a child, you can tell them to go to youtube.com/crashcoursekids and subscribe, and you can go do that as well if you would find that kind of content useful or interesting.